Good afternoon, ladies and gentlemen. I want to welcome you to the Transplantation Innovation Series, really the education series for patients. And I'm privileged to be a co-moderator with Dr. Karen Hechenberger, who is both a physician and a PhD, and is the CEO of Lifebulb. And our panelists today are really a very distinguished panel in every way possible, really. And it's our privilege to have them. I will start with Dr. Gordana Vunjak-Novakovic, who is a university professor at Columbia University and a named professor of biomedical engineering and medicine at Columbia, as well as a member of the National Academy of Engineering and National Academy of Medicine. Dr. Srinivas is the vice president of CareDx and former professor of nephrology. And Dr. Adam Griesemer is professor of surgery at New York University and a distinguished researcher in the field of tolerance induction and xenotransplantation. Our topic for today are several topics, really, is tolerance induction, bioengineering and regenerative medicine, as well as biological markers for rejection, as well as artificial intelligence. As you can see, this is a very forward-looking areas of science and medicine, and really the future, the fantasy world that we are going to be entering it to reality probably in the next 5, 10, 20 years. So without further ado, let me just start. And I think Dr. Gordana Vunjak-Novakovic is going to start and begin this with comments on regenerative medicine.
And before you start, just a few words. Thank you to the audience, and we are definitely paying attention to questions that are coming in. This is a recorded session, so you have the ability to listen to this again, and you can tell your friends to go to transplantlife.com to get the recorded version. And I want to thank you to Veloxis Pharmaceuticals for sponsoring this series.
And right from the start, I want to apologize because I mixed up the order of presentations. But Gordana, go ahead, please.
Okay. So first of all, it's really wonderful to be here. I am a scientist, an engineer. I'm running a research lab at Columbia University. So we love science, but everything we do is really motivated by the opportunities and needs to make some benefit to the patients. So I think these situations, these kinds of meetings are probably the most important for us. I will talk about how we can make progress in engineering human tissues for transplantation. So to this end, I would just like to tell you a little bit about the concept that we follow, and then give you 2 examples so that you can understand where we are in our research, what are the kinds of things that we can do today, where are we going, and where are the challenges. Because not everyone is familiar with tissue engineering, let me just describe briefly the concept using this cartoon. So there are 3 components in every tissue engineering recipe. There are the living cells that are the only and ultimate tissue engineers. Only the cells can make tissue. And we assist the cells in performing their normal biological functions, which is to build the tissue, by two bioengineering components. One is what we call scaffold. So this is a mimic of the normal tissue matrix, providing the framework for the tissue formation. And the other is a culture system or bioreactor, which is providing environmental control, nutrition to the cells, and also regulatory factors. We are instructing the cells on what to do, because starting from the very same stem cells, you can get a piece of bone, a piece of muscle, or a piece of lung. And this is where actually this instruction resides. So you can go towards making a piece of bone, or you can go towards making engineering lung or engineering heart. So now I'll just switch to my examples. Example number one is a project that we started many years ago, which is how to grow anatomically correct, anatomically precisely shaped human bones that can be used eventually for transplantation. So we decided to test our methodology by making the jawbone, because this is one of the anatomically most complex bones, as you can see on this picture, and it is also the most loaded bone in our body. So if we can make something that's anatomically correct and also able to carry this very extensive loading, maybe this methodology can lead us to making other bones in the body. So we would define the scaffold based on clinical images. We would also make the bioreactor chamber for this very complex scaffold in anatomical shape. So the scaffold would fit into this chamber like hand into a glove. You would load the stem cells into the scaffold, and then you would perfuse culture medium through this structure for a couple of weeks, and then 3 or 4 weeks later, you get a piece of living human bone. So we have done a lot of experimentation in vitro and came to the point that we tried to answer question, would this technology work on a clinical scale? And we reached out to one of the most popular models in biomedical research. So this is a pig, because it is human-sized, and then anatomy is similar to great extent, and mechanical loading is similar. So what we have done is exactly what I showed you on the previous picture. Each animal was imaged to get data for making scaffold, for making bioreactor, and each animal donated a small amount of fat. So this was a small liposuction to isolate stem cells and grow a piece of bone. And then when we obtained these pieces of bone, each of which was geometrically and also biologically specific to this particular animal, we replaced a perfectly good jawbone in the pig with our engineered jawbone. And then we looked into how it further develops over about 6 months following transplantation, and we looked also into, I'll show you now, into integration between this implanted piece and the native bone. So what you can see here, this is histology. This is actually section of the tissue that we stain with different molecules so that we can see specific structures. What I'm trying to show you here is something that you can appreciate very easily, which is that the anatomy, the morphology of this bioengineered piece, which has the bone structure, and then you have a transition zone, and then you have a layer of cartilage, is actually very similar to the native bone. The message to take is cells are very smart. You give them some hints about what to do, and they will figure out the rest. And so I just wanted to show you like the result of these experiments. Currently, this technology is I cannot go back. Okay. It is in clinical trial in one of our startup companies. So patients are actually being implanted, and now we're in the second year of this clinical study and really anxiously waiting to see how the outcomes will look in the actual patients that do need replacement of their large sections of the bone in the head and face.
So, Gordana, this bone is made from fat cells, right?
You can make it from
The thing that you started with were fat cells, but it could have been bone marrow cells? Or...
Yeah, it is actually bone marrow. We discard the fat cells. What we use is actually this stromal population, stromal vascular population, which is in fat. So you're using stem cells that come from the fat tissue or bone marrow tissue, or you can use so-called induced pluripotent stem cells that are derived from blood. There are many ways to get cells from the human body. We prefer fat because harvesting is much easier than harvesting of bone marrow, but the end result is pretty much the same.
How do you make sure that you have the stem cells when you harvest the fat?
You separate them, and there are specific tests that you do. For example, you will start from the fat cells, from the stem cells, and you will show that they can make all these different cell types that they make in the body. Fat cell will not do that. Fat cells just is terminally differentiated, and stays what it is. So another thing that happened last year is actually this process that we patented, and this was the basis for EpiBone for the company, actually got some attention, and we got the annual award of the European Patent Office for patents in biomedical sciences and technology. So this was really nice to see. And here I show you a couple photos where you can appreciate how these bones look that we have grown really from scratch from scaffolding materials and the stem cells. So this was my first example. My second example is something totally different. So this is the human lung. And this is an example of that what we do in lab is largely commissioned science. Many of our projects really start from the clinical problem. And this is exactly what happened here. This was about 10 years ago. We were contacted by a group of Columbia transplant surgeons, lung transplant surgeons, who approached us with a problem that they are, as many of you know, they're facing on a daily basis, that there is not enough donor lungs for the many patients that they are at the terminal stage of lung disease and that they don't have any other viable option but the lung transplant. So not enough lungs for people that need them. To make crisis even worse, most of the donor lungs, sometimes even 4 out of 5, are rejected after they are tested before the transplant. They're either not functional enough, there is an injury that was not noticed during harvest, or there is infection or some other reason. So the already small number is decreased further. The field of tissue engineering was attempting to make many tissues, and I showed you a successful example of bone, but lung is just too complex. It has more than 50 different cell types. It has this incredibly complex hierarchical structure of the airway and also of the vasculature. And between the airway, which is this tissue that is facing the airspace and the vasculature, you have about 145 meters square of gas exchange surface. This is what the lung does. It is exchanging oxygen and CO2. And if you don't think metric, this is about half of a tennis court. This is what's packaged in your chest to help you breathe, which is obviously very high on our list of priorities. We cannot make this in lab. Not yet. I'm not sure if we will ever be able to make it. So talking to surgeons, we started to ask ourselves, can we lower the ambition, change the goal, and try to repair those lungs that are rejected instead of making something from scratch? So this became a project that we have been conducting now for about 10 years in collaboration with our surgeons. First step was to start to grow the team, and what we try to do is actually shown on these pictures. What you see here at the top right is our vision of what we may be doing once these lungs are properly repaired. The patient himself or herself who is about to receive transplant may support the lung that they are about to receive for a couple of days until this lung is functionally restored. So to test this hypothesis, we went back to our favorite model and connected the lung to the host animal. So this is again pig. So this lung that you see here is ventilated. It's placed on the respirator, like patients are placed on the respirator, but then it is connected by blood flow with the host. Why is this important? This lung is using liver, kidney, and all other organs that are involved in the metabolism and maintenance of normal homeostasis of the lung of the animal. So you are buying time. You can keep lung in this setting for long enough to do lung repair. How does it look experimentally? What you see here on the left is this dome where we have a lung that we are trying to repair. Under the drapes is the host animal. It is the pig. You see here this big tube. So this is the tube that is connected to a ventilator, and then this lung is connected. You cannot see it very clearly by a vascular supply to the pig, to the host pig. So the blood flows between the two. And then you have a lot of instrumentation that is normally used in OR to monitor the well-being of the organ and of the host organism. I'll just show you two examples. One example is example of a lung. This was a porcine, this was a pig lung that we subjected to severe ischemia. So this lung is all white because this is not really perfused with blood. It's in a very bad shape. You can see here by thermal imaging that these whitish areas correspond to dark areas, which is the low temperature, which is an indicator that there is no blood flow. And then over about day and a half with this regimen of connecting lung to the living host, this lung assume very normal appearance, and this is because they are normally regularly perfused by blood. And then I don't show you much of the data, but actually the structural improvement and the functional improvement always go hand in hand. So what you see here at the left is how we translated this technology to the human lungs. This lung in particular was rejected by multiple transplant centers in the New York area. It was declared a research lung, which means no clinical utility. We get it in lab. We start from like a terrible state of the lung, which is labeled here as 0 hours. And then over time, this lung is improving. And what I want to show you here, this is the only data graph that I'll show you, is the function of our lung is most easily measured by these curves that are called pressure volume. In very simple words, if your lung is healthy, you breathe easily. So you don't need much pressure difference to have a lot of flow. If your lung is in a bad shape, like this lung at the left, you can apply a lot of pressure during inhalation and exhalation, and you still don't have much flow. The lung moved from this very injured state close to this so-called control lung, which is a healthy lung, over the period of 36 hours. And I will just close by mentioning two things that are most important. One is biomedical research is a team work. So this is the lab. These are the people that are really behind everything that we are doing. And the other equally important components are our collaborations. We work with a lot of people that are experts in different areas of science and medicine. And without them, within one single area of science of engineering, we wouldn't be able to make much or any progress. So just stop here, and then I hope we will have enough time for the other two presentations and for any questions that you may have for the three of us.
Thank you.
Thank you. Thank you very much. I think that we'll come back for the questions about repair with cells and repair.
I just want to make one comment, because we're getting this question. A lot of our viewers are kidney transplant recipients or on the kidney list, and just want to say, and I want, of course, to hear your backing me up here, that the repair can be done, and we actually saw that last week at our innovation challenge, that there are systems in place to repair the kidney. If you have a poorly functioning kidney that is coming for surgery, then there are systems already in place and trying to improve that kidney. Is that something that you're working on as well, or are you focused only on the lung?
We are not working with the kidney. We have done some work with liver, but in principle, any perfusable, blood perfused organ, which is heart and kidney and liver and lung and brain, people are also talking about limbs that can be preserved. So in principle, you could work with them. And also, if you saw this very recent example of transplantation between pig kidney and deceased human, this was the same concept. This was also the concept where the two were connected by vascular perfusion. So yes, it's possible, but actually we don't really have any data, any experience with the kidneys.
Thank you.
So now we'll turn to the idea of transplantation tolerance, which is essentially the holy grail of all transplantation, which means really that we can transplant organs without immunosuppression eventually, or with a little immunosuppression at the beginning. And I can't think of any more qualified person to discuss this than our next speaker is Dr. Griesemer, who is professor of surgery and a distinguished transplant surgeon, who focuses on both liver and kidneys, but primarily liver now.
Thank you, Dr. Hardy. To begin with, I just want to define when I'm talking about immune tolerance, what does that mean? So our immune systems are programmed to start to recognize what is part of our body and to attack anything that is not part of our body, because normally those are going to be viruses, bacteria, or fungi. And when we do an organ transplant, there are different proteins and carbohydrates on the surface of a different person's body that our immune systems will recognize as foreign, and they will attack it in the same way they would attack a bacteria. And when we talk about immune tolerance in the setting of transplantation, what we mean is that we reprogram the immune system so that it recognizes the proteins and carbohydrates on the donor's organ as part of our body, but all of its other functions are the same. So it would have the same response to viruses, bacteria, and fungi as before. So we would accept the organ, our immune system would not damage it, it would function its normal lifespan, and we would have a normal functioning immune system to fight off other infections. So why do we focus on doing this in transplantation? Because we have medications that can suppress the immune system, and we can keep our immune system from attacking our transplanted organs. Well, the problem with those immunosuppressants are they're not specific. So they suppress the immune response to everything. So patients on immunosuppression, as you know, have a higher susceptibility to viral infections or bacterial infections. Additionally, these medications, as all medicines do, have side effects. They can promote hypertension. They can promote the development of diabetes. They can promote the development of renal failure. And we know about 10% of heart transplant patients will go on to need a kidney transplant because of the side effects of some of the medicines we use to protect their heart. So ideally, we would be able to do transplants without suppressing the immune system, making people more susceptible to infection, and having the side effects of these immunosuppressive medications. So the concept of immune tolerance and transplant tolerance is actually very, very old. One of the early Nobel Prizes in the mid-century of the 1900s was given to Owen, Medawar, and Billingham for their work on immune tolerance and how that develops. And what they noticed was in free martin cattle, and these are cows that share a uterus, and so they share the uterine blood supply, but they're not identical twins. When they're born, you can do reciprocal skin grafts. So you can take skin from one animal and swap it with the other, and they accept without any additional immunosuppression. So the bone marrow that they shared and swapped in the placenta was the cue for our immune system recognizing what was part of our body and what was not. And this has led to now about 50 years or more of research in performing bone marrow transplants between donors and recipients in an effort to induce immune tolerance. And the concept is, if you can get the bone marrow from the donor to be accepted and survive in the recipient, the bone marrow will provide the cues to the recipient's immune system to reprogram it, to learn that the proteins and carbohydrates on the donor's bone marrow that are also shared with the solid organ from that donor, that those proteins and carbohydrates are now part of the recipient's body. And so the immune system will therefore recognize any organ transplanted from that bone marrow donor as part of its own body, its own self, and it will not reject that organ, while it maintains its normal function in fighting off infections. Now, one of the challenges with doing this in an adult human, or even a child, compared to the free martin cattle were not yet born yet, and their immune systems were totally undeveloped, and they had not developed immunologic memory. And humans, adult humans, have spent a lifetime developing immune memory. And that's why we get vaccines. That is why when we get sick and we get exposed to bacteria or viruses the second time, we're able to fight that off more quickly, because immune systems, immune cells in our body remember that infection. They remember that pathogen, and they survive for decades in our body. And if they are re-exposed to that virus or bacteria, they mount a more rapid response and they fight off that infection. Now, unfortunately, about 1% of these immune cells will also be able to respond to the proteins and carbohydrates on the donor organ. There's some cross-reactivity there. So when we are trying to induce tolerance to the immune system, to organ transplants, we have to reprogram these memory cells and make them forget what they already know. And it turns out that is a very hard trick to do. Whereas the cows that aren't born, they don't have an immune system established. They just have to learn not to recognize those. For adults, we have to get our immune system to forget to recognize those. And that is really the trick. Now, we have successfully done that. And there have been several clinical trials, mostly focused in kidney transplantation with living donors, where they do a bone marrow transplant and a kidney transplant concurrently. The two largest series have been done at Mass General Hospital in Boston and at Northwestern University Hospital in Chicago. The differences between the two approaches was the Mass General group didn't want the bone marrow to survive forever, whereas the group in Northwestern did. And their approaches were a little bit different, because when you do a bone marrow transplant, you can develop a complication called graft versus host disease, where the bone marrow attacks the recipient. And in that situation, from the bone marrow's point of view, its whole body is a transplant. Its immune cells can recognize proteins and carbohydrates in the recipient as foreign and attack those. And graft versus host disease is when that bone marrow transplant attacks the recipient's body. It can give you rashes, diarrhea, it can cause cytopenias, low platelet counts, things like that. And it can be devastating and can be fatal. So the Mass General group was focused on not having the bone marrow survive forever so that you would not develop graft versus host disease. As a result, about 60% of the patients that received the bone marrow transplant and the kidney transplant from the same donor were able to stop their immunosuppression, not have rejection of the kidney, and maintain that graft up. Now I think they have about 16, 17 years from their first recipient without rejection. The Northwestern group did want to get permanent chimerism because, or permanent bone marrow take, because it actually increases the percentage of patients that develop tolerance. The percentage of patients that develop tolerance in that group was over 95%. But they did have a few patients that developed graft versus host disease, and including one patient that I know of at least where it was fatal. So the regimen that was used to induce tolerance actually led to the death of the patient because of the bone marrow transplant. So a lot of research right now is focused on how to do the bone marrow transplant without getting graft versus host disease, with getting the rate of tolerance induction close to 100%, so that everyone that undergoes these protocols will develop tolerance, but will not have the unwanted side effects. So that's what's been done in kidney transplant. It's been mostly focused in that area. One other thing I do want to mention is in liver transplantation, there have been several studies where we have tried to figure out who is spontaneously tolerant. There are a certain number of special patients after a transplant that will develop tolerance with nothing else, no other treatments except standard of care. And we think that's probably more often in liver transplantation than other organs, because when we're very, very young, the liver is actually where our bone marrow cells reside. And in some patients, you do have bone marrow stem cells that reside in the liver forever. So when you do a liver transplant, you're doing sometimes a very, very small bone marrow transplant as well. And in very select patients, we can slowly lower immunosuppression, and in very, very monitored situations and in very highly selected patients, we are able to stop immunosuppression on liver transplant patients without observing rejection at all. We still don't know reliably how to identify the patients that are tolerant, even though we're still giving them medications, compared to other people that if we stop their immune suppression would reject. There are several studies that are looking into biomarkers of tolerance. So what cells and what inflammatory markers in the recipient are characteristic of people who are tolerant, but we don't know, versus others that would reject. So that's an area of active research. And if you are interested in immune tolerance and you have a liver transplant, you can talk to your providers about whether they have any clinical trials. And again, this is at a highly investigational stage. And it's estimated that less than 1% of liver transplant patients can actually come off their medications safely. So don't try this at home. You should only try this in a clinical trial that's highly monitored by your transplant doctors.
So Adam, Dr. Griesemer, just to think about it back again from the kidney perspective, you talked about these two different studies, one being the long-term versus one being a short-term replacement of the bone marrow. How do you see, or how do you explain between living donor and a diseased donor situation? Well,
part of the requirements for those transplants was conditioning of the immune system before the transplant. So there were medications that were given before the bone marrow transplant to condition the recipient's body to accept the bone marrow transplant. And part of that is you have to give medications that get rid of some of your own bone marrow cells. So there's a home for the bone marrow cells from the donor to reside in and to engraft in. So you have to know with a fairly good amount of lead time when the organ transplant is going to take place. And that's really only reliably done with a living donor. And so that was why one of the requirements, at least for the Mass General group, was the living donor was available.
So in the cases of all organs, this would be only possible in liver and kidney?
Yeah, and in primate studies as well, the short-term chimerism only seems to induce tolerance to kidneys, and we're not sure what's special about the kidney in that situation. I did the studies on liver transplant and short-term chimerism where it didn't work, And other groups have done that with lung transplants and heart transplants, and it didn't work. So there was also something magical about the combination of the bone marrow and the kidney when tolerance wasn't permanent. So when the bone marrow transplant wasn't permanent. If the bone marrow transplant is permanent, we think that any organ could be transplanted.
Some people are, and this is something that I think in clinical practice, there are some, especially kidney transplant patients who keep lowering and lowering the medication under guidance, of course, of their provider. Is there a future for them in totally removing the immune suppressive medication at some point in time without having the bone marrow switch?
That's a great question. In general, it's a little bit safer to try that on a liver patient as well, because the liver can regenerate itself. We can take out a huge portion of somebody's liver and it can regenerate. If a kidney is damaged and it loses a nephron, that becomes scarred and you never get it back. So the consequences of failing, taking immunosuppression off entirely for kidney, you will never recover the renal function you had before the failure. It'll always be a little bit less. And for a liver transplant patient, that may not be the case. They may, if you can treat the rejection, they may recover their liver function back to normal. And same for heart transplants and lung transplants. So that's another reason why the majority of these studies have been in liver transplant patients, of just simply taking away immunosuppression. And we are hoping to identify kind of these markers of tolerance and be able to translate some of that knowledge to different organs once we figure it out for liver.
And on that note, we may want to move to our next presenter, but before that, I want to keep in mind for you, Adam, Dr. Griesemer, just to think about, because you talked about a total reversal of tolerance, right, with the bone marrow. But there are also some research going on in more targeted maybe tolerance, and maybe we can talk about that a little bit, afterwards, which is not as drastic.
There's also a number of species where liver tolerance occurs almost spontaneously, particularly in some species of pigs and even rats. So that the liver is a little bit peculiar in that sense. We don't have much time to discuss this, but also the other issue there is the question of antigen in the self and non-self recognition, whether it is possible to fool Mother Nature into not recognizing the foreign antigen. And that is another research option that may exist. Unfortunately, we don't have more time to discuss this for the time being, and so we want to give Dr. Srinivas a chance to tell us about some of the biomarkers and also about the use of artificial intelligence in kidney transplantation and perhaps in medicine in general in the future.
Absolutely. Thank you, Dr. Hardy, Dr. Herdenberger, for the introduction and time. So I'm going to talk about very practical things that are here today. And for those kidney people that are on the talk, that are either have an organ or have talked to somebody in the waiting room who says they're not on immunosuppression, please don't try this at home, as Dr. Griesemer said. Okay. So, where I'm going to talk to you about artificial intelligence is a way in which we use data to make decisions. And, in healthcare, we have a profusion of data. And if you want to define artificial intelligence in a very simple way, it's in a way in which a computer or a device allows you to use them as your human agent and perform some tasks in a way that you would consider smart. And, we all use this every day. If you're going to just get out there in New York City, as I was out yesterday, and you just pull out your phone and decide that you want to walk somewhere, you're using some level of artificial intelligence. This slide that you're seeing in front of you, I just put in a Microsoft PowerPoint, process kind of thing. And I started talking about artificial intelligence and machine learning. And this thing essentially said that if you want to use a computer to perform certain tasks, you need to understand from left to right your data in terms of your disease or your business case, and and understand your disease or business case in terms of the data. Can you make some predictions using some models, which are mathematical ways of looking at data in X and Y? You use X's to predict Y, and algorithms, which are specific sets of tasks that you use in sequence or in conjunction or in combinations to arrive at a certain answer. And then you use those predictions to make some decisions. There's always an amount of uncertainty around this, and you need to be honest about what that is. And then after you make your decision, you learn from it and say, hey, what worked, what didn't work, and just keep getting better at it. We do this over a lifetime. We as humans have evolved. Mankind has evolved from other life forms, and we have done this iteratively. So, and when you do this through a computer, we call it artificial intelligence. So I'm going to just, with apologies, I'm going to say that I'm going to have to talk about my company's product. And we use we use AlloSure, which is donor-derived cell-free DNA. In other words, donor and recipient have separate genomes, and the difference between the polymorphisms in DNA between the two can be used to arrive at a measure of whether the donor organ, which is inside the recipient, is undergoing injury. So it turns out that AlloSure, which is donor-derived cell-free DNA, is very low in stable recipients, the ones that Dr. Griesemer was calling tolerant. It can be elevated at the time of rejection, and it actually comes down after successful treatment. Flip it around. If you want to understand rejection in terms of data and a biomarker, it is one where if there's no rejection, it is the values of AlloSure cell-free DNA are very low. When there's rejection, the value of cell-free DNA goes up, and actually goes up before there's deterioration in function of the allograft. Because by the time you have deterioration of kidney allograft function, you actually have loss of tissue, as Dr. Griesemer was talking about, and you have irreversible scarring. And so you never really get back to normal, you just compensate, and then each hit is worse. So it's good to have this, and when you treat it, it comes down. Data understanding of a problem. So if you have data understanding, can you predict? So one of the first endeavors was for our we worked with the Paris transplant group with the Hôpital Necker and Dr. Lupi and his colleagues. So they used data accrued up to 1 year post-transplant to predict 3, 5, and 7 year graft outcomes. So it's not just astrology, but you're actually using data acquired during the first year post-transplant to predict what happens long-term. And mankind has always been very obsessed with predicting the future, right? And the practical application here is that you can conduct a clinical trial for immunosuppression, get data within this first year, and be able to prognosticate many years in advance without having to do a trial for many, many years. Think about how great an advance that is, right? Because we keep talking about how there are not enough drugs in transplant. So prediction has a role. This is a proof, these graphs are from proof of concept study performed on the Transform trial, which was a large trial conducted by Novartis, where they compared two separate regimens on kidney transplant recipients, and the use of the iBox, or integrated box, which is what this model is called, showed that if you know whether the patient rejected in the first year, you knew their creatinines and their glomerular filtration rates, and you knew their donor-specific antibody, you could predict using 1 year data what 3, 5, and 7 year graft survival was. Great to predict, right? I'd love to know things are going to fail in 7 years, but then we came back to it and said, what's the if you can predict something is going to fail, the immediate question is, what am I going to do today? So it means you have to support decisions meaningfully that change the long-term outcome. Most people will agree that rejection is a meaningful intermediate outcome, and a decision support system that informs the probability of rejection, given some biomarkers and clinical variables, is something that's actionable in the kidney. So we developed something called AI Kidney. And what did we do? First thing we did was to take the algorithm from the Paris transplant group, which used the GFR, or the kidney function at the time of a biopsy, along with an evidence of kidney allograft dysfunction, history of recent rejection within a year, anti-HLA antibody, which is antibody against the donor made by the recipient, and cell-free DNA. And if you look at it iteratively in multiple models, it turns out that when you add all the stuff together, which is the cell-free DNA and all the elements I showed you before, you get a beautiful area under the curve of about 0.85, which means it's 85% accurate in the prediction of whether a patient will have rejection on a biopsy. Think of where this goes from just biopsying everybody with rejection to actually being very focused on how you treat patients in the clinic. Everybody doesn't get a needle in their kidney. So you have an algorithm, it works, and it turns up, and we know it works, but taking that into the clinic is a whole different ball game, right? So what we said was there's two things that we need to know in the clinic. The doctor who's seeing the patient wants to know if the patient has rejection and whether they need to do a biopsy. So it can give you a probability of rejection in real time, which is on the left panel. The patient really doesn't care that much about what's going to happen that afternoon. They want to know what the long-term prognosis is. Could we bring those two together? And so what we did was to pull the biomarker results in the top panel here, as you see, and can be displayed across in time. We bring in the likelihood of rejection score, which tells you whether you should biopsy the patient today or not. At the same time, you're able to have a meaningful conversation with the patient around the long-term prognosis and how the doctor may change some things which may be less toxic to the kidney and put them on regimens that could preserve kidney function a little better now that we know about the toxicities of some of the medicines used in kidney and so on. And more importantly, we doctors like to think we're perfect and our game is on every patient that we see, and that we see all the variables, we understand them, and we make the same decisions every time. It turns out that we're not as smart as we like to think. So AI operating in the background within the EMR, or electronic medical record, and what I'm showing is a mock-up of an EPIC screen, which is what which is the most prevalent EMR across many medical centers is what it can do. So we do submit that we have something here where we have a molecular biomarker that allows early diagnosis of allograft injury, combined with an insight into whether the patient may have rejection and needs a biopsy, combined with the prognostication based on accrued data. And we do provide this in the form of a calculator too. It looks like this. And you put in, for example, you have a 74-year-old male patient, 6 months after transplant, has some instability of the graft function by 2 creatinine measures, a little bit of protein in the urine, modest antibody, cell-free DNA of 0.75, which is just a little elevated. And the biopsy would be predicted to show T-cell mediated rejection. So that's kind of where we are with all of this. And we are expanding this into heart. This is a screenshot from Cedars-Sinai, courtesy of Dr. Jignesh Patel. And they are able to predict whether a patient would have coronary artery vasculopathy and prevent coronary angiograms in heart transplant recipients. And we've been in the business of preventing, heart biopsies for heart transplants for about 22 years now. So the results can be done in a calculator, and we are working on automating this in the EMR. So that's where we are. I'll open up for questions and try to answer them as best as I can.
Yeah, so Srini, thank you. Just a question on the use of iBox. As we have understood, is iBox is conditionally approved in Europe, but we're waiting probably another 2 years for approval in the US. How does that affect this, if at all?
So we are actually excited about that. In fact, you might see a press release come out, just as Veloxis did too. But the main thing here is that it absolutely will accelerate the efforts around this, because surrogacy for a drug is very different for example, from using this in real life as we're doing to guide risk stratification. At the same time, what we're doing here is also to help people do clinical trials or in either their own centers, or even understand the effect of an intervention or a regimen change in a center using the iBox score. So for example, a patient comes into rejection, you can then treat the patient and then check the iBox scores at the time of the start of the rejection and 3 months after, and see what the delta tells you about whether the rejection has been treated successfully or whether the curve of disease has been affected favorably. And could this AI technology be used pre-transplant to predict whether or not the organ may reject? Some folks, there is there is a company out in UK called Verici, which says that they do have an AI platform. I've not seen the results, but they do have it on the website in absolutely full honesty, because there's a non-denominational talk. The leap from pre-transplant to post-transplant is a little bit more difficult, because we would literally have to change the way we deliver immunosuppression up front. And there would need to be accrued knowledge and quite a few clinical trials to say that something done pre-transplant affects post-transplant. And where we are starting is in the post-transplant, and I'm certainly open to the question of investigating pre-transplant, and it's something of an area of active interest for us. And Dr. Hardy, I see that your question and your comment in chat that kind of just contradicted me, but it's complimentary. So,
and
well, we're not quite ready. Unless you do this in vitro, it would be difficult to do, I guess.
Well, the thing is that I'm going at is, if you think about the work that John Rysick and Peter Heger did at Case Western, right? They looked at panel reactive T cells, and with increasing amounts of waiting time on dialysis, the likelihood of rejection post-transplant was higher. So I'm going back to that work, and I'm going to submit that most people confuse panel reactive antibodies as a measure of whether a person is going to reject, but with modern cross-matching techniques and matching, that's actually less of a problem than preformed antibodies, So I think that knowledge is evolving, if I may submit respectfully, that we may need to think about this a little differently than we have,
So can I just try to bring it back to practical life for a patient and a doctor? For both of, actually 3 of you, and to a certain extent Gordana as well, but the 3 of you who are practicing physicians, seeing patients, this test, this specific test, when would you use it? In the patient's, when you see the patient? And right now it's only indicated in kidney, as I understand.
No, the AlloSure test. Which one are you talking about? Which test are you talking about?
The AlloSure.
Okay, right now, so we have AlloSure for kidney, we have AlloSure for heart, we have AlloSure in development for lung and for liver. So we have all of these going on. So we have these across all organs. AlloSure is available for heart, and we do have expression profiles also available.
So when would you use it for the patient? How often? When?
So we typically recommend that you can do it anytime after the first 2 months, and we do it every month for about 6 times in the life of the allograft in the first year, and then quarterly after that. The test has a very high negative predictive value. In other words, if it's negative, it speaks to something that's quiescent in the allograft, whereas if it starts inching upwards, especially in the context of the other parameters that I said, then the likelihood that a person has rejection is higher, and higher values portend more severe forms of rejection. And after successful treatment, the values come down, which allows you to follow the patient. So
if it's positive, you go forward with a biopsy?
Yes, ma'am. And if it's negative, we advise people to look at other reasons for why the patient could be having problems.
And it's very useful in determining when a rejection has been successfully treated and reversed.
Correct. And we are learning much more about this because we're now going to be tying it up with transcriptomic data as well. so that we're not just looking at a marker of injury, which I will submit. I mean, although most injury that is persistent in an allograft we know is alloimmune, but it is a great mark. It tells you, it follows a time course, but we would like to understand more in terms of the transcriptome as well, to further get away from this whole reliance on histology, which has been going on since the time of Anton van Leeuwenhoek and Richard Bright, right? We need to get beyond that. And so that's kind of where we're going with this. One of the other parts that we need to keep in mind is that this is a lead indicator. So AlloSure or cell-free DNA goes up before the creatinine goes up. By the time your creatinine goes up, 50% of nephron of the kidney has probably been affected by a process, right? So getting to kidney before there's loss of tissue is a big deal.
Yeah.
As Dr. Griesemer said, in liver you can get away with a little bit, in kidney you can't, you know.
So Gordana, let's turn this for a moment to your treatment of and repairing lungs or repairing organs in general. Do you feel that actually, I would imagine that under these circumstances, in fact, I know, you can treat poor blood supply or lungs that are waterlogged, you can dry them out. You can treat them with antibiotics if they are infected before putting it into a recipient. But what about if there's actually a tissue damage? Or is there now thoughts about placing some lung-oriented cells in the lung to fix small segments of lung or big segments of lung?
We have done this, Mark. We have repaired mechanical physical damage to the lung by removing damaged cellular material and replacing it in the same session with the cells that have capacity to regenerate either epithelium or the vascular cells. So obviously there are limits because lung, as we all know, has this ability to self-repair as liver does, assuming the lung is healthy. But then you do need structure. This structural guidance is important. So if the damage is massive, there is not much you can do. If the damage is localized, then you can really do something. And this is this fine line, because what we have learned from the transplant surgeons is these lungs that are rejected, they're not completely dysfunctional. They're just not functional enough. And sometimes we are just talking about maybe 10% or 20% of functionality that is missing. And this is something that is in many cases within reach. But not every lung can be repaired using this methodology.
So would you agree or comment about the fact that, for example, repairing the heart may be much easier than repairing a lung or a kidney because of the variety of cells, the location of cells? And
I think all of this, all three organs that you mentioned, nothing is easy. And it just varies.
Well, Adam told us last time that the heart is the easiest to get from a pig. So I remember that.
It's difficult because heart is different. I mean, it is a little bit like kidney, which is that the damage tends to stay permanently. Cardiomyocytes in particular, they have zero capacity for renewal. So this is the, people sometimes ask, like, how come you make bone, you cannot make lung here or heart? Here is the answer. You know, if the tissue itself has very large capacity for self-repair, your job is much easier. It gets more difficult with multiple cell types, but then stem cells come into the picture. So I just want to say this is still at the research stage, and I think we are not close, except for bone, to going into patients. There are many, many things that need to be determined, including we are looking closely into immune aspects. Obviously, this is a xeno situation. like a human organ and an animal. And even with these genetically edited pigs that are more immune tolerant, many challenges of the immune incompatibility remain. So Adam was talking about it already in great detail.
Well, we're coming to the end of this, so I just want to thank all the panelists, the speakers. It was most exciting and stimulating discussion. I'm sorry that we have to end it because there's much more to say. And obviously, as you can see, each one of our speakers can probably speak for a week about their subjects and still not exhausted and would keep us interested. So thank you very much for participating and for sharing with us your forward-looking ideas and concepts. And I thank my co-moderator, Dr. Hegenberger, and I want to thank Veloxis for supporting this effort. And Karen, you have the last word.
You know, I'm not gonna. I'm really impressed with how you communicated these strong, scientific data and the future. But what was really compelling to me was that how the three of you are addressing very different issues, and that combined, it really could be part of the future for patients with transplants. We need better markers. We need to use better markers. We need better regulatory pathways to actually be using those markers. That's why I was pushing the iBox. But also we need to get more supply for the very high demand that we have. We need to regenerate or repair or, do xeno. I think we need to improve our drugs. And the drugs need to be improved from where we are now to, of course, that very drastic improvement of bone marrow transplants, but there's something in between as well. So, the future of transplant, I think, is bright. We have seen it today. We saw it in the previous seminar, and I want to really thank our audience as well for staying with us and having such great interaction with us. So thank you so much, and we'll see you very soon again.
Very good. Thank you so much. Take care.
Thank you. Thank you.
Thank you.